So pyruvate must first be converted into acetyl CoA before the citric acid cycle, also known as the Krebs cycle, can begin. And this is mediated by a major enzyme complex called pyruvate dehydrogenase complex, which I'll refer to as PDH. So within this reaction, pyruvate and coenzyme ASH are substrates that end up producing a molecule of carbon dioxide, acetyl CoA. A molecule of NADH is also produced during this process, and this protein complex mediates the conversion of pyruvate to acetyl CoA. To complete this conversion, there's 3 enzymic processes that have to occur. First, there's a decarboxylation of the pyruvate molecule to release the carbon dioxide, and there's a triacetylase reaction that moves the acetate functional group of pyruvate to coenzyme A carrier to produce acetyl CoA. So during this process, electrons are harvested from the substrates and transferred from an NAD plus carrier to form NADH. And this redox energy harvesting step is a dehydrogenase reaction, and the enzyme complex is named for this very important step in the reaction. So PDH has 3 active subunits called or known as E1, E2, E3. The reactions performed in the PDH complex have several specialized chemical cofactors that are used by the enzyme subunits. So these cofactors include thiamine pyrophosphate, also known as 2-PP, lipomide, flavin adenine dinucleotide, also known as FAD, and nicotinamide adenine nucleotide, or NAD+. The E1 enzyme subunit, which performs the first step, uses thiamine pyrophosphate, or 2-PP, to extract some carbon dioxide from the pyruvate. So this first step is known as decarboxylation. And at this point, the second pyruvate combines with the ionized form of 2-PP, and this is then decarboxylated to produce hydroxyl or 2-PP, and this is catalyzed by the pyruvate dehydrogenase component E1 of the PDH complex. And then the next step would be the oxidation. At this point, the second cofactor, lipomide, becomes involved. The hydroxyl group attached to the 2-PP is oxidized to make acetyl group, while being simultaneously transferred to lipomide, which results in a thioester bond. And so during oxidation, acetyl lipomide is also produced. So once the acetyl group has been attached to the 2-PP in the E1 subunit, the oxidized form of the lipomide cofactor enters the subunit. The enyl carbon in the 2-PP intermediate structure has an ionic character, enabling it to attack the disulfide bond of the oxidized lipomide cofactor, and this releases and restores the TPP cofactor in the E1 subunit and opens the ring structure of the lipomide to create a reduced thiol functional group, and the other sulfur loaded with the acetyl group from pyruvate. The E1 catalyzed process is the rate-limiting step of the whole pyruvate dehydrogenase complex. So here we can see partially reduced lipomide cofactor forming a thiester, and the intermediate acetyl group from the pyruvate. So the acetylamide intermediate, so we're gonna talk about this step now. So the intermediate would be this. So this intermediate will then shift from the E1 subunit and move to the E2 subunit. And in the E2 subunit, or active site, the reaction transfers the acetyl from the lipomide to the thiol of the coenzyme A, and it produces acetyl CoA, which is released from the enzyme complex and enters the citric acid cycle. So in the fourth step, the oxidized form of lipomide is regenerated by dihydrolipoyl dehydrogenase, also known as E3. So once the dihydrolipoyl has migrated to the dihydrolipoyl or the E3 active site, it undergoes a type of oxidation. So first, FAD oxidizes dihydrolipomide back to the oxidized lipomate state, producing the reduced FADH2. So the FADA cofactor that cannot be released from the enzyme. So to make electrons more available for use in the electron transport chain, the electrons are transported to the mobile NAD plus cofactor. So NAD plus oxidizes FADH2 back to the FAD resting state, producing NADH and hydrogen atoms. So NADH and the hydrogen atoms can then leave the PDH enzyme complex and shuttle the electrons to the electron transport chain. And at this point, the enzyme is reset and it's completed one round of the catalytic activity, producing one molecule of coenzyme A, one molecule of carbon dioxide, and one molecule of the NADH and the hydrogen atoms. And so this reaction will happen twice for each glucose molecule process from the glycolysis. Thank you.